Hello students, in today's class we will be studying the process of double fertilization. It is the most unique and an important process which is exclusive to angiosperm. So before understanding the process of double fertilization, let's just recall the structure of embryo sac that I have told in my previous video. So this structure is of embryo sac. It has one end, two opposite poles. One end is called as the micropyle and opposite to it is a chalazal end. At the micropylar end, we have three cells, two synergids with finger-like projections called as piliform apparatus and one egg cell. Then we have two polar nuclei in the center of polar cell, and we have three cells at the chalazal end which are called as the antipodals. Okay, so this is the structure of the embryo sac. Now let's recall about the pollen grain. So in majority of the angiosperm, in 60% of the angiosperm, the pollen grains, they are shed at the 2 cell stage. That means it has one vegetative, or you call it as tube nuclei, and one generative nuclei. So in the pollen pistil interaction, I told you that when the pollen tube, it is in the style, that time the generative cell, it divides and it forms two male gametes. So this happens when the pollen grain are shed at the 2 cell stage, and if it is shed at the 3 cell stage, already there is one vegetative nuclei and two male gametes. Now let's recall what happened in pollen pistil interaction. The pollen grain, it landed onto the surface of the stigma. Then there is a recognition mechanism occurred to find out whether the stigma has to accept or reject the pollen grain. That happened by identifying whether it is a compatible or a non-compatible pollen. If it is a compatible pollen, then the pollen tube growth started. First started with the pollen germination and then the pollen tube growth. Then the pollen tube is guided towards the micropyle, and that is because there are chemical signals which are released. There are chemicals which are released from the filiform apparatus which guides the pollen tube to enter through the micropyle. So that is how the pollen tube enters, through the micropyle end. So now the entry is to one of the synergids. So the pollen tube, it starts entering from one of the synergid, and that synergid we call it as receptive synergid. This path is guided by the filiform apparatus, remember, and it because it provides a chemical signal. Okay, so the pollen tube, it enters through one of the receptive synergids, and soon this synergid will be degenerated so that it will create a space for the pollen tube to release the male gametes. Okay, and with that, two male gametes will be released. Soon the another synergid, this will also degenerate. Why? Because this will ensure that only a single pollen tube enters inside the embryo sac. Now what is left? It is the egg cell. It is only the egg cell which is left. Now we'll understand what happens during the double fertilization. So now comes the process of double fertilization. Double fertilization is a process where two fusion events take place. Okay, the first one is called as Syngamy. Syngamy means when one of the male gamete, it fuses with the egg. So the male gamete is also haploid and so is the egg cell. Now this fusion forms the zygote, which is diploid. So the first fusion is called as syngamy, which is the fusion of male gamete with the egg, and it forms the zygote. The another fusion is called as triple fusion. That is another male gamete, or you can call it as the second male gamete. It fuses with the two polar nuclei. So the second male gamete is also haploid and the two polar nuclei are also haploid, and this forms the primary endosperm nuclei, which is triploid. Okay, so double fertilization, we call it as a double fertilization. Why? Because there are two fertilization events that are occurring. One is the syngamy, fusion of one male gamete with the egg, and the another is a triple fusion, which is the fusion of the second male gamete with the two polar nuclei to form the primary endosperm nuclei. Now sometimes these two polar nuclei, before it fuses with the male gamete, it fuses with each other and it forms a secondary nuclei, and then the second male gamete fuses with the secondary nuclei to form the primary endosperm nuclei. Okay, so next, whatever event that is occurring after the zygote and the primary endosperm nuclei is formed comes under the post fertilization event. So in the post fertilization event, the zygote develops into the embryo and the primary endosperm nuclei further divides to form the endosperm. Okay, the role of endosperm is that endosperm, it provides nutrition to the developing embryo. So endosperm is always formed after the embryo has formed. After the first syngamy happens, then only the triple fusion occurs. Okay, so the zygote will develop into embryo and the primary endosperm nuclei will divide and form the endosperm, which provides nutrition to the developing embryo. So in my next video, I will be talking about the post fertilization events, that is the development of embryo and endosperm. So stay tuned, and if you like my video, please like, share and subscribe. Thank you.